Chapter 7 of Honkera for Histology 8.3 Cartilage. Cartilage is a tough, durable form of supporting connective tissue characterized by an extracellular matrix or ECM with high concentration of GAGs and proteoglycans interacting with collagen and elastic fibers. Structural features of its matrix make cartilage ideal for a variety of mechanical and protective roles within the adult skeleton and elsewhere. Cartilage ECM has a firm consistency that allows the tissue to bear mechanical stresses without permanent distortion. In the respiratory tract, ears, and nose, cartilage forms a framework supporting softer tissues. Because of its resiliency and smooth, lubricated surface, cartilage provides cushioning and sliding regions within skeletal joints and facilitates bone movements. As described in Chapter 8, cartilage also guides development and growth of long bones, both before and after birth. Cartilage consists of cells called chondrocytes embedded in the ECM, which, unlike connective tissue proper, contains no other cell types. Chondrocytes synthesize and maintain all ECM components and are located in the matrix cavity called lacunae. The physical properties of cartilage depends on electrostatic bonds between type 2 collagen fibrils, hyaluronan, and the sulfated GAGs on densely packed proteoglycans. Its semi-rigid consistency is attributable to water-bound to the negatively charged hyaluronan and GAG chains extending from proteoglycan core proteins, which in turn are enclosed within a dense meshwork of type 2 collagen fibrils. The high content of bound water allows cartilage to serve as a shock absorber, which is an important role or functional role. All types of cartilage lack vascular supplies, and chondrocytes receive nutrients by diffusion from capillaries in surrounding connective tissue, the perichondrium. In some skeletal elements, large blood vessels do traverse cartilage to supply other tissues, but these vessels release few nutrients to the chondrocytes. As might be expected of cells in avascular tissue, chondrocytes exhibit low metabolic activity. Cartilage also lacks nerves. The perichondrium is a sheath of dense connective tissue that surrounds cartilage in most places, forming an interface between the cartilage and the tissue supported by the cartilage. The perichondrium harbors the blood supply, serving the cartilage and a small neural component. Articular cartilage, which covers the ends of bones in movable joints, and which erodes in the course of arthritic degeneration, lacks perichondrium and is sustained by the diffusion of oxygen and nutrients from the synovial fluid. As shown in Figure 7-1, variations in the compositions of the matrix characterize three main types of cartilage: hyaline cartilage, elastic cartilage, and fibrocartilage. Important features are summarized in Table 7-1. Okay, Table 7-1. Hyaline cartilage, mixed feature of ECM. This hyaline cartilage is homogeneous with type 2 collagen and aggrecan, while in elastic it also is type 2 collagen, aggrecan, but a darker elastic fiber. In fibrocartilage, as to type 2 collagen as well, with large area of dense connective tissue with type 1 collagen. For major cells in hyaline cartilage, you have your chondrocytes and chondroblasts, as well as in elastic cartilage. For fibrocartilage, you have your chondrocytes and fibroblasts. Typical arrangement of chondrocytes in hyaline cartilage: they are isolated or in small isogenous group. In the elastic cartilage, usually in small isogenous group, and in fibrocartilage, isolated or in isogenous group arranged axially. Presence of perichondrium. In hyaline, yes, it is present except in epiphysis and articular cartilage. For elastic cartilage, it is present but not present in fibrocartilage. Main locations or examples for hyaline cartilage: its many components of upper respiratory tract, articular ends and epiphyseal plates of long bones, and fetal skeleton. For elastic cartilage, you have your external ear, external acoustic meatus, auditory tube, epiglottis, and certain other laryngeal cartilages. For fibrocartilage, you have your intervertebral disc, pubic symphysis, meniscus, and certain other joints, insertions of tendons. For main functions, your hyaline cartilage provides smooth, low-friction surfaces in joints, structural support for respiratory tract. For elastic cartilage, it provides flexible shape and supports soft tissue. For fibrocartilage, it provides cushioning, tensile strength, and resistance to tearing and compression. Medical application. Many genetic conditions in human or mice that cause defective cartilage, joint deformities, or short limbs are due to recessive mutation in genes for collagen type 2, the aggrecan core protein, the sulfate transporter, and other proteins required for normal chondrocyte function. Hyaline cartilage. Hyaline cartilage is the most common of the three types. It is homogeneous and semi-transparent in the fresh state. In adults, hyaline cartilage is located in the articular surfaces of mobile joints, in the walls of larger respiratory passages like the nose, trachea, larynx, and bronchi, in the ventral ends of the ribs, where they articulate with the sternum, and in the epiphyseal plates of long bones, where it makes possible longitudinal bone growth. In the embryo, hyaline cartilage forms a temporary skeleton that is gradually replaced by bones. Medical application. Osteoarthritis is a chronic condition that commonly occurs during aging. It involves the gradual loss or changed physical properties of the hyaline cartilage that lines the articular ends of bones in joints. Joints that are weight-bearing like knees and hips or heavily used like wrists and fingers are most prone to cartilage degeneration. Fragments released by wear and tear to the articular cartilage trigger secretion of matrix metalloprotein, proteinases, and other factors from macrophages in adjacent tissue, which exacerbates the damage and causes pain and inflammation within the joint. Matrix. The dry weight of hyaline cartilage is nearly 40% collagen embedded in a firm, hydrated gel of proteoglycans and structural glycoproteins. In routine histology preparations, the proteoglycans make the matrix generally basophilic and the thin collagen fibrils are barely discernible. Most of the collagen in hyaline cartilage is type 2, although small amounts of minor collagens are present. Aggrecan, which weighs 250 kilodaltons, with approximately 150 GAG side chains of chondroitin sulfase and keratan sulfate, is the most abundant proteoglycan in the hyaline cartilage. Hundreds of these proteoglycans are bound non-covalently by linked proteins to long polymers of hyaluronan, as shown schematically in Figure 7-2A and discussed in Chapter 5. These proteoglycan complexes bind further to surface of type 2 collagen fibrils. Water-bound to GAGs in proteoglycans constitute up to 60 to 80% of the weight in fresh hyaline cartilage. Another important component of cartilage matrix is the structural multi-adhesive glycoproteins chondronectin. Like fibronectin in other connective tissues, chondronectin binds specifically to GAGs, collagen, and integrins, mediating the adherence of chondrocytes to the ECM. Staining variations within the matrix reflect local differences in its molecular composition. Immediately surrounding each chondrocyte, the ECM is relatively richer in GAGs than collagen, after causing these areas of territorial matrix to stain differently from the intervening areas of interterritorial matrix. Chondrocytes. Cells occupy relatively little of the hyaline cartilage mass. At the periphery of the cartilage, a young chondrocyte or chondroblast have an elliptic shape with a long axis parallel to the surface. Deeper in the cartilage, they are round and may appear in groups of up to 8 cells that originate from the mitotic division of a single chondroblast and are called isogenous aggregates. As the chondrocytes become more active in secreting collagens and other ECM components, the aggregated cells are pushed apart and occupy separate lacunae. The cartilage cells and matrix may shrink slightly during routine histologic preparation, resulting in both the irregular shape of the chondrocyte and their retraction from the matrix. In living tissues, chondrocytes fill their lacunae completely. Because cartilage matrix is avascular, chondrocytes respire under low oxygen tension. Hyaline cartilage cells metabolize glucose mainly by anaerobic glycolysis. Nutrients from the blood diffuse to all chondrocytes from the cartilage surface, with movements of water and solutes in the cartilage matrix promoted by intermittent tissue compression and decompression during body movements. The limits of such diffusion defines the maximum thickness of hyaline cartilage, which usually exists in small, thin plates. Medical application. In contrast to other forms of cartilage and most other tissues, hyaline cartilage is susceptible to partial or isolated regions of calcification during aging, especially in the costal cartilage adjacent to the ribs. Calcification of the hyaline matrix accompanied by degenerative changes in the chondrocytes is a common part of the aging process and in many respects resembles endochondral ossification by which bone is formed. Chondrocyte synthesis of sulfate gags and secretion of last sentence, the limits of such diffusion define the maximum thickness of hyaline cartilage, which usually exists as small, thin plates. Chondrocyte synthesis of sulfated gags and secretion of proteoglycans are accelerated by many hormones and growth factors. A major regulatory of hyaline cartilage growth is the pituitary-derived protein called growth hormone or somatotropin. This hormone acts indirectly promoting the endocrine release of the liver from the liver of insulin-like growth factors or somatomedins, which directly stimulates the cells of hyaline cartilage. Medical application. Cells of cartilage can give rise to either benign or chondroma or slow-growing malignant or chondrosarcoma tumors in which cells produce normal matrix components. Chondrosarcoma seldom metastasize and are generally removed surgically. Perichondrium. Except in the articular cartilage of joints, all hyaline cartilage is covered by a layer of dense connective tissue, the perichondrium, which is essential for the growth and maintenance of cartilage. The outer region of the perichondrium consists largely of collagen type 1 fibrils and fibroblasts, but an inner layer adjoining the cartilage matrix also contains mesenchymal stem cells, which provides a source for new chondroblasts that divide and differentiate into chondrocytes. Elastic cartilage. Elastic cartilage is essentially similar to hyaline cartilage except that it contains abundant network of elastic fibers in addition to a meshwork of collagen type 2 fibrils, which gives fresh elastic cartilage a yellowish color. With appropriate staining, the elastic fibrils usually appear as dark bundles distributed unevenly through the matrix. More flexible than hyaline cartilage, elastic cartilage is found in the auricle of the ear, the walls of the external auditory canals, the auditory or eustachian tubes, epiglottis, and the upper respiratory tract. Elastic cartilage in this location includes perichondrium, similar to that of most hyaline cartilage. Throughout elastic cartilage, the cells resemble those of hyaline cartilage both physiologically and structurally. Fibrocartilage. Fibrocartilage takes various forms in different structures, but is essentially a mingling of hyaline cartilage and dense connective tissues. It is found in intervertebral discs, in attachments of certain ligaments, and in the pubic symphysis, all places where it serves as a very tough yet cushioning support tissue for bone. Chondrocytes of fibrocartilage occurs singly and often in aligned isogenous aggregates, producing type 2 collagen and other ECM components, although the matrix around these chondrocytes is typically sparse. Areas with chondrocytes and hyaline cartilage are separated with other regions with fibroblasts and dense bundles of type 1 collagen, which confer extra tensile strength to this tissue. The relative scarcity of proteoglycans overall makes fibrocartilage matrix more acidophilic than that of hyaline or elastic cartilage. There is no distinct surrounding perichondrium in fibrocartilage. Intervertebral discs of the spinal column are composed primarily of fibrocartilage and act as lubricated cushions and shock absorbers, preventing damage to adjacent vertebrae from abrasive forces or impacts. Held in place by ligaments, intervertebral discs are discussed further with joints in Chapter 8. Cartilage formation, growth, and repair. All cartilage forms from embryonic mesenchyme in the process of chondrogenesis. The first indication of cell differentiation is the rounding up of the mesenchymal cells, which retract their extension, multiply rapidly, and become more densely packed together. In general, the terms chondroblasts and chondrocytes, respectively, refer to cartilage during and after the period of rapid proliferation. At both stages, the cells have basophilic cytoplasm rich in RER for collagen synthesis. Production of the ECM encloses the cell in their lacunae and then gradually separates chondroblasts from one another. During embryonic development, the cartilage differentiation takes place primarily from the center outward. Therefore, the more central the central cells have the characteristics of chondrocytes, whereas the peripheral cells are typically chondroblasts. The superficial mesenchyme develops as the perichondrium. Once formed, the cartilage tissue enlarges both the interstitial growth, involving mitotic division of pre-existing chondrocytes, and by appositional growth, which involves chondroblast differentiation from progenitor cells in the perichondrium. In both cases, the synthesis of matrix contributes greatly to the growth of cartilage. Appositional growth of cartilage is more important during postnatal development, although as described in Chapter 8, interstitial growth in cartilaginous region within long bones is important in increasing the length of these structures. In articular cartilage, cells and matrix near the articulating surface are gradually worn away and must be replaced from within. Because there is no perichondrium to add cells by appositional growth. Except in young children, damaged cartilage undergoes a slow and often incomplete repair, primarily dependent on cells in the perichondrium, which invade the injured area and produce new cartilage. In damaged areas, the perichondrium produces a scar of dense connective tissue instead of forming new cartilage. The poor capacity of cartilage for repair or regeneration is due in part to its avascularity and low metabolic rate.